Hello, everyone. Good evening. Good evening to everyone, all of you. I welcome one and all to our session. Am I audible? Yes, please confirm in chat if I'm audible to you all. We apologize for the slight delay, but I can assure you that the sessions that we are going to have today is definitely going to compensate for this 5 minutes delay. I promise you that, right? So without any further delay, we'll jump on to beginning our session for today. Hello, once again, I am Urmimala and I will be your host for today's session on how to empower your biological research with data science. We learn about the tools and the techniques. You see, in the last few years, we have witnessed an incredible shift in biology. You will all agree with me, yes or no? Right? So earlier, research was mostly in labs, all about experiments, hours spent in labs. But today, biology has become data heavy. It is driven by next gen sequencing, multi-omics data, clinical studies, and massive environmental data sets as well. So here's the reality. The modern biologists can no longer ignore computational approaches. Whether you talk about disease modeling or ecological trends or CRISPR target validation or even cancer research, data science has become the language of discovery. Agreed, right? So, what does this mean? It means that every modern biologist now needs to think like a data scientist. And don't worry, that doesn't mean that you need to become a hardcore coder or mathematician overnight. It simply means learning the right tools and approaches to make sense of that massive biological data which we have around us. So, in today's sessions, we'll help you all to do exactly that. So, this is how we have structured the session. First, we'll explore how biology has transformed into a data driven field and why it is urgent for researchers today. Then, we will introduce the essential tools and platforms you can start using for your research, and even if you don't have a coding background. And trust me, this special session is going to be very important for you because we have a special guest with us here today. We are very fortunate to have her here today. And finally, we will guide you step by step on how to begin your data science journey in biology and build the skills that can open up careers in research, biotech, and even beyond. Finally, we will end with a live Q&A session. So stay with us till the very end to get your questions answered directly by our experts. But first, let's welcome our esteemed speakers for the day who will guide us through today's session. So first up, we have Ms. Shubhi Singh with us today. Shubhi Singh
Thank you,
is
ma'am.
a dedicated Shubhi Singh is a dedicated researcher and educator, having completed her PhD work in biotechnology from SRM Institute of Science and Technology. She brings a strong interdisciplinary background and seamlessly blends computational and experimental research. Her expertise spans molecular docking, admit prediction, protein ligand interaction analysis, and bioinformatics workflows for target identification and drug design. She is also skilled in integrating in silico modeling with wet lab techniques such as qPCR based gene expression profiling, microbial culture under stress conditions, and biofilm inhibition assays. Alongside her research pursuit, Ms. Singh has significant experience in academic teaching and laboratory supervision, where she fosters scientific skills and critical thinking among students. Her work is driven by a commitment to advancing research in antimicrobial resistance and therapeutic development through innovative data driven approaches. Thank you for joining us today, Shubhina. Now, for our second session, we are proudly honored to welcome our esteemed speaker, Dr. Nupur Mukherjee. Welcome, ma'am.
Good evening, everyone, and thank you for having me here. And among such intelligent people, I am a doctorate in AI in sustainable pharma, and please make it very free flowing. I'd like to also learn from you and share you a very industry perspective of what we are doing in the world of pharma, biostatistics, and research, and few anecdotes for you to remember in very easy ways. Right. So, thank you for having me, Umala.
Thank you, ma'am. We are honored to have you here rather today. To brief about Dr. Mukherjee, she's a global technology leader with over 27 years of experience driving AI first digital transformation across industries, including biopharma, healthcare, and life sciences. Dr. Nupur currently serves as Agentic AI Competency Head at GlaxoSmithKline, where she leads global enterprise AI strategies accelerating drug discovery and development through cutting edge agentic AI solutions. Her leadership has delivered around $4 million in savings while scaling AI driven innovations across 68 countries. She has previously held senior leadership roles at some of the world's top organizations, including Standard Chartered Bank, Barclays, HSBC, WMWare, Accenture, Capgemini, and Wipro, bringing a unique cross industry perspective to data science analytics and AI implementation. An AI and sustainability expert, Dr. Nupur holds advanced certifications from Stanford University, Oxford Saïd Business School, and the Johns Hopkins University, along with a recent honorary doctorate for her work in AI driven sustainable innovation in pharma and banking. Dr. Nupur has authorized white papers on AI in drug discovery and has been instrumental in developing scalable AI powered solutions for clinical data, ESG compliance, and bioinformatics applications. Once again, it's a privilege to have you with us here today, ma'am. Thank you so much for joining, and we are all eagerly waiting to hear from you.
Thank you so much.
Next
The
up,
honor is
we
mine.
have with us our very own Yes, ma'am, most welcome. So next up, we have with us Mr. Prodyot Banerjee, who is a seasoned professional specializing in bioinformatics, computer-aided drug design, and genomics with extensive research experience at premier institutes, including CSIR IGIB, CSIS CLRI in Chennai, IIT Madras, and Delhi Technological University. Sir holds an MTech in bioinformatics from DTU and has qualified national level competitive exams including CSIR and GATE, reflecting his strong academic foundations. Mr. Prodyot has a robust research portfolio with 8 publications in reputed journals such as IEEE and Frontiers in Pharmacology, accumulating a cumulative impact factor of 43.84 with more publications underway. He has also presented his research at top platforms like IIT Kharagpur, highlighting his engagement with cutting edge scientific discourse. His deep expertise in bioinformatics analysis, drug design, and genomics, combined with a proven track record in R&D, makes him a very valuable contributor to the fields of computational biology and precision medicine. Thank you for joining us today, sir.
Thank you for having me. It's a pleasure.
Likewise, sir. So now that we have met our speakers, it's time to dive right into today's sessions. I would like to invite to the platform our first speaker for the day, Ms. Shubhi Singh, to start us off. Over to you, Shubhi ma'am.
Yeah, thank you so much, Urmimala ma'am. I will just quickly share my screen and start the session.
While ma'am is on sharing her presentation, why don't you tell me who do we have in the audience today with us today? So, what are your backgrounds from? Are you a student, currently a student, or a working professional, or a young researcher? I would like to know from you all. Let me know in the comment section, and it will be very nice for our speakers to customize their sessions accordingly for you. Okay, so Shubhi ma'am is ready with her
Ma'am,
presentation.
ma'am, my slides are
Yes,
visible?
Yes, your slides
Am
are
I
visible,
audible
ma'am.
properly?
Yes, you're audible, ma'am. Please go ahead.
Okay. Yeah. Thank you so much, ma'am. So I'll go ahead with my topic. So good evening, everyone present over here. So in my today's topic, I'm going to just tell you everybody about the biological data explosion and are we human ready for it or no? Okay. So here, so you must be asking, you must be thinking, what is biological data explosion? So in the past, we used to work with, like in the history, I mean to say, we used to work with like one protein or one gene or like one pathway. Okay, that like we used to work step by step. Okay, and it used to take us a lot of time to work on a one particular thing. Okay, if it's a protein, if it's a gene, or if it's a pathway analysis, and we when it comes to the wet lab proving, it used to take a ample of time for us to prove it in the wet lab. Whereas nowadays we have a lot of advanced techniques such as NGS. Yeah. So we have a lot of advanced technique such as NGS, that is your next generation sequencing. Here, using NGS, we can sequence billions of data DNA bases in an hour. Whereas in the earlier ages, it used to take billions of year or maybe like decades of year to sequence the data. Okay, next we have like transcriptomics, proteomics. Transcriptomic is basically like we can within an hour or so we can get to know about like thousands of gene what is present in the whole sequence, thousands of protein what is present in the whole sequence, and we can measure all those things. Whereas in the field of clinical data also and in the in medical technology also, we use a lot of AI and ML in these fields also. Okay, if we say, so this is about the biological data explosion, comparing to the data from the past to the future, what we are doing it. Next comes, are we like really ready for the biological data explosion? What is the thing and what it is about? So there are like so many challenges are there, and like the humans are really humans are ready for the biological data explosion, and what is the thing? So here we can say like there is a lot of sequencing techniques are there. As I have mentioned earlier, there's a lot of sequencing techniques are there, and in that we see like we perform the sequencing in a very fast note. Okay, we don't take like really much time. It just take like about an hour. Either we go for the Illumina sequencing, either we go for the nanopore sequencing. It just takes a very less time compared to the earlier. Okay, so like
Shubhi,
if
ma'am,
we use
sorry for interrupting. Sorry for interrupting, but your slides are not changing, ma'am. If you could share
yes
the full
ma'am.
screen presentation.
Ma'am, I'm just still on the first slide only, ma'am.
Okay, okay, okay. Thank you for
Yeah.
confirming. Yeah, sure.
Yes, yes, ma'am. I'll be moving ahead. Yes. So, yes. So, are we like really ready to take up all these things? Okay. So, let's take a recent example of like maybe human genome project. So, human genome project. So, human genome project maybe started a long time back, but it took up a lot of time to complete because it just started in like 90s and it just got completed by 2000 or something. So because taking all these things into consideration, that is why we need a biological data explosion. Okay, so from experiment to data. Okay, so here, as we can see, the first point what it says like biology before and experiment heavy and hypothesis driven. What it says over here is biology used for the experimental purpose and the data hypothesis driven. Okay, so mainly we use the biology in such a way where we will have like a wet lab validation with it. We will have a hypothesis based on that, and then we try to prove that hypothesis either with the wet lab or with the dry lab. Okay, so biology is such a way it has been come up to us. Next comes biology today. So earlier time, what we used to do, we used to hypothesize something and we go for the experimental way and we used to get the result. But now what in today's world, biology is more of a data heavy and discovery drive. Okay, so here the term data heavy and discovery driven, what it says. So data heavy means nowadays we are mainly dependent on the AI and ML, that is your artificial intelligence and machine learning. So we are looking into artificial intelligence, machine learning, and what just happens, like suppose we have a very huge databases with us, so it's very difficult to sort the data manually. Instead of sorting the data manually, we go with the machine learning techniques or the artificial intelligence. With that, we can sort the data easily, and we can present it in a better way, or we can, it gives us a quick result also compared to doing it manually, right? So next comes, like suppose you just take, like for this, if I give you an example for like discovery driven and data heavy, is like we have a lot of just suppose you have a transcriptomic data, okay, and transcriptomic is basically identification of the genes in the data, or it's basically for the protein identification in the whole sequence. Okay, so now we have the transcriptomic data. If the humans start to sort by one that data, it takes up a lot of time. Whereas if we have a lot of machine learning, or maybe like a lot of artificial intelligence algorithm, where we give the input file, it sort our data based on, suppose we want to identify the cancer related genes, it sort our data based on the cancer related genes, and it gives us the output. So that is what is our today's biology. So data is now a first class citizen in biology. Okay. So what I can say is, like in the past, we used to have data as a byproduct. Okay. We used to use like in silico method or all these things as a byproduct for the biology. We used to mostly rely on the experimental method. As I told, experimental method is like a very expensive method, and it takes up a lot of time. But in fact, if we see, human don't have, like nowadays we human don't have that much of time and patience. So that is why we only had to come up with something, with the new things, where we can where the experiments can be done quick and the results can be given very quick. So we came up with the data, the AIML and by AIML and machine learning, and that makes our job very easy. Okay. Next comes, if I take the example of sequencing, you just take an example of sequencing, like the genome sequencing, where you do the identify, like where you sequence the whole genome, okay, whole DNA basis. So what just happened earlier days, where we had, when we didn't have a proper genomic uh, machines and every other thing, so it used to take us a lot of time, it used to take us like years and years. But nowadays, as we have all the facilities available, the sequencing work just gets over within an hour or so, and the data interpretation, we can just finish it within like you just take like 2 to 3 days. If you give it properly, you can finish up the data interpretation, and you will have the all the data present in front of you. Okay, so that is the transition from the experimental data in the history, from the today's date, where we use AI, ML, and machine learning. Okay, so next comes, we have like different types of biological data today. Okay, so what it says about. So different biological data today is, first one, if I talk about, is the next generation sequencing. Why I keep talking about the next generation sequencing, because it's like most of the audience is familiar of this topic. Okay, mostly people are aware of DNA RNA sequencing, those who are like studying in, from the, those who are from the biotech background or from the biology background. Even in the 12th 10th and 12th standard, we have just come across these terms. So that is why I'm keep repeating about the NGS, and I'm giving you as an example for the NGS only. So types of biological data, first we can see what is NGS about. So what here it does, it generates DNA and RNA sequencing, okay, and it generates the data in a massive scale. That means it will give you like the data of 10 GB, sometimes 10 to 15 GB. Okay, so here we can what we can see over here is, if we have the whole genome sequencing of either of a DNA, either of RNA, that is your transcriptomics. Here we can find out a lot of genes. Suppose we have a huge data and we want to find out the cancer genes related to cancer, or maybe like we want to find out the gene related to Alzheimer, any of the disease, we can take it, and then we have a ML pipelines. We go over there and then we run the data and we get to know like how many genes are responsible for causing like certain type of cancer from the whole transcriptome. Okay, so that makes our work very much easier. Next comes your proteomics and metabolomics. So proteomics stands for the proteins, okay, and the metabolomics stands for the metabolites. So metabolites such as your carbohydrate, lipids, all these things. So what here we look for? So if we see, it's the easiest way to identify the biomarker for the disease. Okay, well, like we have a different biomarker for the different disease, like your Alzheimer or your like cancer is a widely most known disease, like everybody must have heard of this term, so that's why I'm telling about the cancer. Okay, so these like we can use it as a biomarker, and then we can compare it with the biomarkers at the protein and the metabolic level using the proteomics and metabolomics. Next comes here our clinical data. So what it is clinical data about? So here, like it includes the patient health record, okay, like your imaging techniques such as your MRI, CTC imaging, okay, and it will be helpful for the diagnosing, and it will be helpful for the pathology, and like the any other things if we are probably looking for from the clinical data, what kind of tumor it is, what the like suppose we take what the medicines are prescribed to this patient, so there it's your clinical data. Okay, next comes your omic integration. So what is omic integration? Omics is basically the when we combine everything together that forms the omics. Everything in the sense is there: genomics, transcriptomics, epigenomics, and metabolomics, and and even the proteomics. If we combine all the terms together, it gives us the omics integration. So here, omic integration, what we do, suppose let's take the example, we have like a cancer research. In the cancer research often we use for the genomics. Okay, we look for the in the genomics we look for the mutation, where the mutation has occurred in the particular genome, and where the which particular gene is getting mutated. Okay, so we look go for the genomics. Next comes like your transcriptomics we look for the gene activity, which gene is probably upregulating, which gene is downregulating. Okay, then comes your proteomics is all about the proteins, which protein is getting like some protein sometimes what just happen, it will be expressed at a gene level but once we go for the protein level it won't be expressed. So we look for the at what level the at the proteomics level the certain proteins are expressing or no. Okay, so these are the few terms which we use it for the biological data today. The last ones is our environmental data. So what is environmental data and how environment affect our biology? Okay, so this is all about environmental data. So it includes like your microbiome. Okay, microbiome such as you can take gut bacteria. Okay, you can take ecology, the climate effect. Okay, the climate effect is one of the main thing which affect the human health a lot. Okay, so if I give you an example, suppose you just take like uh, microbiome studies that shows the gut bacteria influences obesity or maybe the immunity in the human, or even sometimes it affects mental health also in the human. Okay, so next, we can see what are the problems basically the biologists face. Okay, so the data is there, but we don't know how to use it. Okay, that's the biggest problem I get to hear from the biologists. Okay, those students who are not uh, like they just come up to me and they are like, "Mom, we have a lot of data, but we don't know how to work with that data." Okay, so that's the biggest problem nowadays the biologists are facing. So today, there are different labs which generates a different huge amount of data. Suppose I told you, like in the field of genomics, we generate like around 10 to 12 GB of data. Okay, sometimes we generate the terabytes of data. So there are different labs that generates a lot of huge amount of biological data, such as your proteomics data, genomics data, and every other thing. But many biologists, they don't have the computational training to analyze it. Okay, so that is, the computational training is very much needed for the analysis of the data. So that is why we are looking for, like, to teach a lot of students to learn about the coding and the basics of coding and the biopython and every other thing, so that the unused data, the unanalyzed data, we can look into that and we can analyze the data. Okay, so like we are just the researchers are just keep analyzing the data, they just keep generating the data, and we are keep submitting into the databases. But those data, what is the output from that, that is sometimes it's not known to the world, like it's not represented in the paper form, or it's not published anywhere. So that is why we need a biologist with a good technical knowledge and with a good AIML knowledge to analyze those data. There are many challenges we face while doing this process. So it's like, we can, first is, too much of data is there, but we do not have the enough skills. So like, suppose you have you have a lot of sequencing data, but we don't have the skilled labor to analyze those data. Okay, next we look for the lack of integration between the biology and the computing. So what just happened, like those who are into the wet lab, or it's like those who are working on the benches, they are much more comfortable to work in the labs. Okay, and they are not really aware of how the computer works, how the AI and ML get integrated into the wet lab data, and that gives us that makes our jobs easier. So big, that is one of the other reason we face the challenges in that. Next comes choosing the right tool and right algorithm. So we have a lot of tools for one. Suppose we want to do a one particular analysis. Okay, well, let's take, like, we want to do admit analysis. We have a lot of tools, but we don't really know, like, which tool is how much is it is accurate, and which tool we should go and use it, and what is the algorithm behind the tool. So that is one of the other problem which we face in this. The last one is need for the collaboration with the data scientists. So now it's not compulsory that all the scientists, suppose if I'm a, so you just take, if I'm a wet lab person, okay, I work on the bench, it's not compulsory for me to know each and everything. I should not, it's not compulsory that I should know the computational part also, I should know the wet lab part also. Okay, so that is why I go for the collaboration. Suppose I'm good in wet lab, I'll go find, talk to my colleague, those who are good in dry lab, or those who are good in computational work. So I'll go and collaborate with them, and that makes that will be the with this way, the both will be benefited. I also will be benefited, and my colleague also will be benefited with this, because if we are going for the publication, we both will have the authorship in the paper, if we look into that aspect. So there we just face these challenges while stepping into the big data explosion. Okay, next comes the biologists always be in a dilemma. Okay, the data is there but don't know how to use it. That's a very common thing which everybody looks for. Data is everywhere, but we don't know how to use that data, how to do, what to do with that data. Like, it's a very huge problem for everybody. So as I told, like, what usually the scientists or the other researchers do, they sequence the data. Suppose you go for the genomics, you sequence the data and you go and submit it into the databases. Whatever you need it, you take it for you, and then rest of the thing you just leave it undiscovered. You just go and submit it over there in the database, and it will be undiscovered. The people will not be aware, the students or the other researcher will not be aware what this data is about. So that is why it's always a very big question mark what to do with the data, and I don't know how to use the data. Okay, so traditional is like, if we look in the traditional biology, like traditional biologists were more comfortable with the experimental data, like growing cell, working with the cell line, doing molecular work such as a PCR, running gel, and doing the performing the microscopic technique. But now as I told, experiment generates a huge data, and that need to be analyzed and should be present in a proper way. So for this way, we need a we need a skill, and we need a different tool. That is why we need a skill labor in the biology also. So as we see, like we are shifting towards the AI. So this is a very big question mark, and this is one of the highly driven field which we are going. So why this matters? Why do we need to have a biologist, skilled biologist? Why do we need to analyze the data? Why this all this data explosion is important? So first one, if we look from here, okay, so first we look for the wasted resources. Okay, so wasted resources here, what I am trying to say is like the if we go to the wet lab, it's like a expensive experiment. Wet lab is always an expensive experiment, okay, and it generates, like suppose we do a wet lab and then we go for the genome sequencing, and after the genome sequencing we will have a terabytes of data or the gigabytes of data. Okay, but if we don't analyze that data, the investment is totally on the whole genome is waste. Okay, next comes data collected but not analyzed. So as I told, the data will be there, the labs will produce a lot of data, but the data will be unanalyzed, it will be left out with the question mark only. And the other researchers, they need to, if you really are in a need of the particular type of data, you will not have a proper answer or maybe the proper analysis for the data. Okay, next comes our missed discoveries. So what is missed discoveries? We'll look here for. So missed discoveries is like valuable biology insight that remain always hidden. Okay, example, if we take a cancer biomarker, maybe it might already be in the existing data, but it never get discovered because we just keep dumping the data in the databases. We don't try to analyze the data. Okay, next comes the hidden pattern remain buried. Okay, so what it is about? So without computational tools, complex pattern in the genomics, transcriptomics, or in the proteomics, that remains always invisible, that will not be shown up to the world. Then poor reproducibility of data. So what it is about? So science depends always on being able to repeat and validate the results. Okay, if data isn't processed well, the reproducibility of the data will be difficult, and the mistrust will come for the data. Next is the results can't be validated. So raw data without the proper analysis means the conclusion cannot be checked, and the conclusion will not be proper if it's not validated properly. Next come the reliance on other. So biologists without the computational skills must depend heavily on the data scientists. So that is why we need a data scientist over there. And this delay research and limit the independence. So if you already know about the AI and ML, if you're aware of this, because then you will not be relying on anybody else to process your data, and that could save up a lot of time for the scientists to work. And that is what about your delay research in progress. Next comes the real world biology research powered by the data. So, what is real world biology? Okay. So, first we can see like different terms like a disease modeling, ecology trends, CRISPR target validation, okay, and microbiome health. So first we'll start with the disease modeling. So what is disease modeling? What it means? So disease modeling is mainly uses multi-omics data. Okay, multi-omics means transcriptomics, genomics, proteomics, metabolomics, and all. And along with the patient clinical data, it to understand disease progress and predict the response of therapy. So it predicts the progress of the patient, it predicts the response of therapy also in that. So if we take the example for this, so in the cancer research, if we take, so AI models trained in omics, okay, and it trained in patient history. So omics and the patient history, when both comes together, the treatment will become the best. The treatment which we give it for the patient, that will become the best for an individual. And this is what is the foundational for personalized medicine. Okay, so why does it matter? You must be thinking like, why does it matter? Why do we have to do the disease modeling? Like, why it matters? Because one side fit all. There's a term. So what it says, the therapies, we can either tailor treatment plan, okay, we can improve patient survival, and we can reduce the pain or the drug given to the patient. Okay, next comes the ecological trend. So, what is ecological trend? So, researchers, they usually combine satellite imaging data, okay, that is from the environment, climate, or they take it from the land use or whatever it is, okay, and the genomic data. Genomic data, that is from your plant, animal, or microbe, maybe if you take it from there, and we combine both the things to study the ecosystem, at what environment this plant will grow, what kinds of genes are upregulating at this environment. So we look for all these things and we go for the ecological trends. And why does this again is like, why does it matter to us? And this helps the conservation biology, and it will guide us for the protecting of endangered species, and it maintains the ecosystem health. Next comes our CRISPR target validation. CRISPR nowadays, it's a very well-known technique which all the scientists and almost all the researchers are working on it. So CRISPR-Cas is used for the gene knockout screening, and it systematically disable the gene and it observe the effect of the gene. Okay, so why do we need to do this? So if knocking out the gene if knocking out the certain type of gene, if we kill the cancer gene or the cancer cell but spares the healthy one, that genes become a drug target for us. Okay, like we have a CRISPR-Cas9, through that we go and we look like, okay, these are the particular genes which are responsible for causing cancer. So we try to eliminate those genes only. We try to kill those particular genes without disturbing the healthy cell or without disturbing the any other gene. So there we use the CRISPR target validation. Okay. Next comes our microbiome human health linkage. Okay, so here what we do, we look for the metagenomics. We include the metagenomics with the clinical record, and that will always influence us to that will influence us to get to know about the chronic diseases which happens in human. Like as I told, from the gut microbes, it might affect your human brain, and it might affect your human health also. So it is about the microbiome health linkage data. Next comes the data led discoveries and the impact of it. So what is data led discoveries and what are the impact of it? So data led discoveries will have the impact in genetics, in the cancer research, in the pharma field, and in the public health research. Okay, so how it affects is the genetic. Genetic means like by sequencing the genome, okay, and analyzing the big say data set, researchers can pinpoint the mutation responsible for causing a particular disease. Suppose there is a mutation in the particular genome. With the whole genome sequencing, we can get to know where the mutation has occurred, and we can try to take out with the help of CRISPR. If we say we to try to knock out that particular gene, and we with that the disease can be cured. Next comes the cancer research. So cancer is not one disease. Okay,
Shubhi
it
ma'am, we are sorry
varies
for
from.
interrupting. Yeah, sorry for interrupting, Shubhi
Uh,
ma'am. We
pardon,
request you to
pardon, ma'am.
please wind up
Yeah,
within
yeah,
two
yeah,
minutes.
yeah, yeah,
Yeah,
yeah,
thank you.
sure. Sorry. Yeah. Yeah, so we look for the cancer research. Okay, so here in cancer research, it varies from patient to patient, and it integrates the different data like genomics, proteomics, and the metabolomics. Next comes to the pharma. So pharma is like we have a traditional drug discovery is a slow and the expensive process. Okay, so we go for the AI-driven drug discovery. In that, it will help us to find out the target and the inhibitor or maybe the drug against it. So we AI/ML drug discovery driven approach. It helps us to find out the target very fast and find out the drug for the particular target. Next comes the public health. So public health we can include as a epidemiological data, okay, where the genomic surveillance will be there and the AI model will be generated. Everything will be included in the public health. And next, we want to know the importance of data awareness before using the all these two. So we have spoken earlier, the data, the awareness about the data should be proper, and the tools which we are going for analysis of these data should be very important. So first, we look for the data quality matters. Okay, so for understanding or analyzing the data, the quality of the data really matters. It should be all proper. There should not be like extra here and there, other fields should not be there and that. We need to analyze the data properly, then we can go for the experimental check. Next come ethics and privacy. Okay, as we are the biologists, we should be ethical towards our work. We should have a proper ethics to work in the field of research. If we are not going with the ethics, the upcoming researchers, the upcoming scientists, they will not have the proper data for them to work ahead. So that is why we consider ethics as one of the foremost important things for for the researcher. It's a very most important thing. Next comes the context first, tools later. So what it is about, tools are not just a magic. Okay, so we have like a lot of algorithm, we will have a lot of fancy algorithm, we have a lot of types of algorithm with which we have the best for the what type of data we need to look for that. So that is why we look for the tool, we look for the best tool to analyze the particular data. Next comes the last one is the better decision. Okay, when the researchers are aware of the data limitation, they can make the accurate prediction. They ensure the results are proper and the reliable, and the reproducibility of the result should be there, and the data should be processed carefully. Okay, so these are few of the things before we go for these are about the data awareness and what kind of tool we need to use for the analyzing the data. Yeah, so this is all about my lecture. So thank you everyone for the attention. Thank you very much.
Thank you so much, Shubhi, ma'am, for the clear and relatable picture of how biology has shifted into the data era and why every biologist need to be data aware. That was truly eye-opening. So next,
Thank you, ma'am.
let's move to the exciting part, the tools and platforms that make data-driven biology possible. Please help me welcome Dr. Nupur Mukherjee, ma'am, once again, as she will be sharing her insights on how to leverage data science and AI to revolutionize life sciences and healthcare by taking us through the essential tech stack every biologist should know. Over to you, Dr. Nupur, ma'am.
Thank you, ma'am. Can we also have questions in between so that because I won't be able to see the screen, would that be fine? Because I'm going to make it very, very light-hearted, and like I said, that like every Indian parent, when I was a small girl, young and beautiful, at that time, my mummy said that you are not the brightest kid in the block, right? And my endeavor throughout my entire life is to make very, very complex things simple. Now, how many of y'all in this audience have played laser tag? Any hands, emotions? Okay, so if you are playing laser tag and you're in this game, so there is an icon when you hit somebody, you seem that the device on your chest and your back decides to vibrate and you know you're out. So that is the basis with which we are now using to detect data. Secondly, is that however brilliant you are, once you're in a company or you're doing a research, remember something, that there is two concepts that will hit you really hard. I'm sorry, it's breaking your heart, but this is the fact of it, right? Because you want to land in like a pro, or as in Hindi we say, don't want to be a kacha limbu. You want to come in as a tenured person. So always remember that whenever you're doing your studies, there's a concept of ENPV, EROI, means what is the net present value of the outcome that you're going to get from the research, right? Number one. Secondly, you're going to look at, after this particular drug or after this particular research is created, then what is the return on investment I'll get. So now what happens is, it's like somebody suddenly calling you, and especially for the women in this group, imagine you're coming home tired through Bangalore traffic, and suddenly your family informs you that, guess what, 20 people are going to come and you're going to fix dinner and Swiggy is not working, right? So now in earlier days we used to start to cook. Now we have Swiggy, we have Zomato, right? So the way you will calculate that, whether out of those 20 people are vegetarian or not, whether they have allergies or not, what type of snacks, dinner, lunch that you will give, and also you want to find out what are the discounts on Swiggy. That is the exact way that we do biostatistic studies, cell plasticity, protein segregation, protein locking. Like for example, I have a small child. If I say that, would you like to have paneer? And she'll say, no, mommy, I'm vegan. So I have to get tofu, right? So just like that, you have to go ahead and just like how you order food for guests, that's exactly how you have to do. And I'm going to give you one really very path breaking way of how things work in real life once you enter the industry. So that said, I'm going to show, let me know if you're able to see my screens.
Yes, ma'am, it's visible.
Okay, perfect. So this session that I'm going to do is going to be a very practical way of how you are going to make it through innovative AI. And let me tell you for granted that I am not starting as an engineer. So I came in from the domain because I interned at various places. So my work at Johns Hopkins was on oncology and Alzheimer's. I interned as a lab statistics person, then in Stanford Medical. And that's why I would honestly say something that you will not get on LinkedIn or on ChatGPT or anywhere else, is that your domain matters. But then along with domain, how can you get your AI power is the magic word, right? So what is the main thing, as my esteemed colleague earlier said, is that what you really need to do first is to start with the data in mind. What is the data stack you have? But just like I talked about, you playing laser tag, do you want AI to work for you or you to work for AI? I would like AI to work for me, right? So what we do nowadays is that, as a rough order of magnitude, uh, you know, if you don't mind, I would like to go in this mode so that I'm able to see people. Are you able to see the screens right now? Yes,
Yes,
no?
ma'am, it's visible. It's visible.
Okay, perfect, perfect. So please feel free to chime or send some emoticons if that's. I know you're listening and I'm not boring you to that. So now what happens is that earlier we were told to find patterns. Now you remember in the beginning I mentioned two terms, ROI and ENPV. The easiest way to bring down the timelines is to take your entire data set or even chunks of the data set or sample and plug it into a GPT or an AI system and ask the AI system in simple language, like a prompt. I will share those prompts after this session. That think you're a biostatistician, find patterns in this data. This will run for a few hours or minutes depending upon how many tokens you have. But what you get is this. You get your hypothesis, right? You also save on manual analysis. You also find out what is the patterns of the data. You also look at publication bias. This is a really shortcut way, because when I started with BERT and GANs, it used to take me around 17 hours. Do you know how much time it takes now? Any guesses? 27 to 34 minutes. Super, no? So that is one of the ways in which you can have this data driven approach. You can also find out if there are multiple
Excuse me, ma'am.
hypotheses and how. Yes,
Excuse me, ma'am. Sorry to interrupt. Can you please put it on full screen, ma'am?
sure. Is this
Yeah,
better?
thank you, ma'am.
Yeah,
Yes, thank you. Thank you, ma'am.
yeah. So what happens ideally is that you can get multiple hypotheses, you can find the correlation of data, and there is something called as tree of thought. So for example, now you and I are talking about data, we are talking about AI, we are talking about pharma. Suddenly, if I say what is the score, what is the average, will I be talking about pharma or cricket? It will be talking about pharma, right? So it helps us keep to context, and as a result, large scale data sets can be plugged in, processed in the matter of minutes, and we can get system level insights without us having to write a single line of code. Not required at all. Now, with GPT-5 getting launched last month, we don't need that anymore. This is already about me, so I'm not going to spend too much of time. You can find me on LinkedIn. Now, what I would again say, don't let the world tell you that just because there is AI, your skills don't matter. Your skills are going to be the difference, and it's the gold mine by which you will get top notch employment going forward to the next 5 years, because a lot of the tech is getting automated. The people who will still stick on are the people with the domain. All you have to do is to join the dots. Now, from just tools to how can you have career opportunities either as a solo entrepreneur or as a person who's probably doing analytics, and there will be a lot of work out there. Guess why? Simple reason is that one of the areas, what are the two areas for highest amount of AI hallucination? Just guess. It's bio use cases, and second is astrology. So by learning this, either you will be an excellent biologist, biostatisticians using AI, or the other career option is astrology. Now, how do I make sure that there is employability in AI biology is how we will look through this particular setup. Now, the first is, I know it's a little detailed, but I'll try to encapsulate it into totally 6 areas. And these are 6 areas of specialization. First, you will start as a generalist who's AI in pharmaceuticals. Please follow the laser pen so that it will step to context. So in AI, we have basically 5 areas. One is as ma'am said, we have the protein structure analysis, genomic data processing, and network biology, where a whole lot of people in India are involved. They are involved in hubs like in Nagpur, in Mumbai, in Hyderabad. And Hyderabad, we have Pharma City. Now there's something starting up in Coimbatore as well. And there is statistical modeling. A lot of these biostatistics, as my esteemed colleague correctly said, is now getting completely automated. However, the research and development, I would really request you to follow what the Belgium model is all about. If you see that they are letting humans decide it, because there is so much of hallucination or wrong data being given by AI. And then thereafter there is research, and as my esteemed colleague will follow me on his journey, you will see that why this human thought, human creativity, human critical thinking is so important. So in the first part, the biology, and you can see the research part, you will see a lot of human involvement. But the areas where you will have to know what AI is doing and also moderate it, govern it, is biostatistics and biochem. Because enzyme kinetics, molecular interaction, drug target binding, these were painfully manual processes that used to take months, years. Average is 17 months. That we've reduced to 3.5 months to 4 months before it goes to in vivo or in vitro testing. And workflow integrations is part of, there are softwares available, so probably you don't have to learn this area. But I would say that if you're really looking at your career, then look at automation solutions that are out there in terms of biostatistics and biochem, and we will see two very robust solutions that you can learn as a newbie in this particular area. So like I said, there are 6 areas for you to develop. But for you as a domain expert is biology applications, especially in this hierarchy, any sort of transformation that you're doing. And insights, so I already showed you that first let the data work for you. This is the detailed diagram. Probably if later on you want to use this for reference, this is going to be your mind map that we use always as a get out of jail sort of model. And then finally, how do I get started as a domain expert, right? I presume nobody knows Python, nobody knows MATLAB, nobody knows anything about code. They've not written a line of code. So the first 4 elements, we call it the Panchamahabhuta, right? Air, wind, fire, earth. First is the visualization layer, because guys, whatever doesn't sell and whatever you can't show, it's just a lot of documents. And right now in the world of Instagram and X, nobody has that much of time to read, so you have to sell your point. Then is the analysis area, or what we call as insights layer. Then the data layer, the infrastructure layer, and the fifth part, which I call it as the magic masala, is that your own insights and competitive difference. So you also have to do a little bit of a reconnaissance, or what in Indian language we call it Reiki, to understand what your competitors are doing, and always provide that particular angle of it when you are providing your research window. That why am I doing this? How is my output coming in? So the difference between a pharma domain AI expert and you, because you've attended this call and taken your valuable time, is that you will provide that by doing this, this is the return on investment, this is the time to completion that I'm doing. Comparatively, other projects will take 2 years. I will be able to complete this in 7 months. So you have to show that while you're going forward. Now, going forward, we have visual tools for non-coders like PyMol, and I will show you also how the PyMol works. It shows you like the molecular visualization for 3D structures. So you know which molecule is related to each other, protein structures, your publications, are there any existing publications? And my friends, knowing PyMol is very important. In the initial stages, if you go to Google, you will see there is a lot of critique on PyMol that is not scalable, yada yada. But when you are starting in your career and when you're doing the use cases, without understanding the visualization that PyMol shows you, you will not be able to make it very further in the career. So you have to show that structural representations, and PyMol is a accredited tool that various FDA in the US accepts, and you'll be able to see the 3D insights for biologists. So let me ask you one question. Uh, why is ciprofloxacin given for brain related injuries and inflammation, and not any other antipyretic or inflammatory drug? Any answers? The PyMol will tell you very clearly that ciprofloxacin is an oil based medium which will be able to penetrate and optimize brain cells, which a normal antipyretic or anti-inflammatory will not help, because they are water or hygroscopic based. Now, if somebody says that for cancer targeting, you would have certain genes that do not. So, for example, somebody's got pancreatic cancer or anything else, then in that case you will be able to say that few drugs do not work. Like for example, if somebody has uterine cancer, then in that case there are certain drugs like Jemperli that work because the binding is there. That is where the magic is. That's where you earn the money. That what are the binding sites and molecular interactions, and how you can induce and change them, and discover new drugs or variants of it, right? So this is providing a graphical image, and it at least one single graph saves at least 200, 170 to 200 pages. So it is well loved by approvers. If you want to patent it, you would definitely put in a PyMol chart. Now, guess what? PyMol charts also, as a rudimentary, can be done by something called as mermaid.ai. It takes $20 a month, but you can get a very similar, very rudimentary base setup. But I would honestly say, please learn this. I learned it. It made a huge, huge difference. The other part of it is cytoscape. So whatever is on your left side is like your holy book, right? You have to make sure that you know this, you have worked on it, and this visualization and interaction is done. Otherwise, biostatistics, the way we are doing it in the industry and globally, that will you will still be an outsider. This is what we've been doing for the last 18 months across industries, right? Now, the other part of it is that once you got savvy with PyMol and Cytoscape, and you understood the regulatory networks, the interactions, etc., uh, you would have to understand some sort of powerhouses, especially on the RNA sequence analysis. Because I remember when I went into a job, the first thing they asked us to verify or work on is the RNA and testing. If somebody gives you testing, please don't worry, because a testing person will be able to see the data from bottom up, and there is no better way to learn. And there are some data gold mines called Geo and NCBI datasets. But guess what? You can also look at AlphaFold. There are few elements of AlphaFold that is available in Gemini 2.3.5, not Gemini Banana. This is Gemini 2.3.5, and in GPT-5. If I were you, I would still go for GPT-4, but you can try both of these options, and you can also create synthetic data should you want it. Now, I hail from the great state of Maharashtra, and there is a very powerful leader called as Shivaji Maharaj. And they used to ask him that, how did you form this kingdom? And he used to say, Shri Checha. Now, I never thought that he was talking about scalable practices, reproducibility, and interoperability. If you are doing any solution through your domain, through AI, which is not scalable, you cannot replicate it, and interoperability is not there across various use cases, or even protein synthesization or epigenomics, then it won't be approved by the AI council within the R&D industry. So as a result, always whenever you're thinking of any solution, tell that you're following the SRI or SRI framework. These are practical ways of you. If you talk like this, people will think you're a tenured person, and as a result, this will not, you will come across as a very landed person rather than a newbie. Now, like I said, that why did I plug in my basic data set into the machine first? Is because this new patterns that you are seeing out here, the machine themselves will provide. Only you have to tell the machine to think like a biostatistician. Then you have to use these words, that do novel hypothesis generation, and say, can you find some efficient handling of biological systems when you're writing your prompt or your question? And you will find that the system will automatically do that. One trick that I used, okay, because I was hard pressed for time, I used that, and I said that whenever I'm trying to do the data analysis, as my colleague earlier said, it is also for a targeted disease. I will not run a data set for common cold and lung cancer in the same go, right? Agreed with me? So in such a case, I will have to give target prioritization, outcome prediction, and say that, you know what, I want to reduce my trials. So when you're looking from these slides, especially from slide 9, slide 10 and slide 11, please put these in your prompt, like literally copy paste it and then run the data. You will find that there's a high level of accuracy. It will take a little bit longer, as a few more minutes, depending upon your size of data, but you will find that you will save on months of research, and all you have to do is to validate the data. Now, I have given a very quick flow. First is you're building a data stack, right? You saw the data storage, processing, data analysis, visualizations, etc., right? So first is that how am I going to get into, I'm a biostatistician, I'm a biology person, I'm a research person, and a brilliant person, but how will I kind of analyze it? So it's your data skills that's very makes you indispensable. And more importantly, people want you for your domain. People want you to see patterns which no one else has done. Right? So as they say, na kabhi kabhi kit kit karna is a very good thing. Look at patterns, try looking at faults that no one else has seen. On the other side, if you look at PyMol, what does it do? It helps you to understand the 3D molecule without the code. Right? So it's like understand Bangalore's metro, which yellow line goes where does green line go? Now, why is it so important for my friends here from MSc students is because it is a drag and drop. I don't have to do too much of rocket science at all. And on the cytoscope, what happens? It's an open source platform. So as a result, there's also a very, very intelligent setup. Like when we go to ArcSiv and get white papers, or IIIE, we get papers, the think tank is there. So as a result, you will not be working on something that is, or a protein strain that is already got discovered. So it saves a lot of plagiarism time, lot of wasted time on or duplication of work. Now, right now in the industry, we use this PyMol cytoscape and a much more higher end systems. I'm not entitled to disclose that for competitive reasons. We use this in team reviews. You will not have this in your studies, but it is vital to present your findings in these two setups for pitching and getting the funding for your use case. You will not get funding otherwise. And as a result, you will reduce your trial and error. And like I said earlier, you have to say that, traditionally, if I do this, it takes 2 years. You do it in this recommended way, as Nupur ma'am was saying, or how you have found out, then it's going to take 7 months. So please give me funding for it. And you will see 99% of the times you will get funding. Last but not the least is how exactly will you approach it? So first is the PyMol and cytoscape, like I talked about. One word of gyan, or what we call as golden nugget, is that in addition to all of these, you have new emerging AI like GPT-5, AlphaFold, which is again a much more higher version of PyMol. But start with PyMol so that you can get into AlphaFold. DeepMind, which is a lot of data. DeepSeek. Now, DeepSeek is you have to go through a Claude and also ask specific data with hallucination controls. But it can generate your hypothesis, summarize your scientific literature in a very big way. We have used AlphaFold sometime for protein structuring on few of our drugs that got released. And there is a wide amount of algorithms that are available in DeepMind and DeepSeek, right? Especially on drug discovery. So now, without boring you further, so we are talking about the protein ligand interaction inside. Guess how much time it took? This actually took around 45 minutes or so to kind of develop. Can you all tell me what is this picture of? For those who are biostatistics people? This is a picture of the variations of common cold or influenza fever that is present in the world today as of April 2025. So if somebody says I got COVID or I got flu, so you can choose out of these colorful dots which one of them have you got. Now, it also lowers barriers for you to get accepted. Number one, it is empowering domain experts. You're not saying that I'm a techie or not techie. It's also making sure that you don't have to depend on technical people for your own data analysis. And some of these tools provide very drag and drop interfaces. So I don't have to wait for a coder to get assigned and you explaining pharma to them or explaining biology to them. You are holding the steering wheel of your own research car going forward. Right? Now, very quickly, you have bioconductor packages, which is DESQ or HR. They will show gene expression data accurately. Now, while you're learning this, remember something: every pharma, every bio research company has their own bioconductor packages. The packages also do any sort of differential expression. Differential expression means how one gene is working very, very significantly to other. I'll give you an example of differential expression. So some of us took COVID vaccine, different types of vaccine. The effect of the vaccine after the vaccine was different on people of different epigenomics, and as a result different demographics, different racial groups. So how do you understand that the response took place across, and what is the impact, what is the the symptoms? I'm just over mansplaining it. That is, if you want to do it, and especially want to launch a drug, you have to also make sure that you know how the entomology of the drug is going to be looking at. And like I said, that you remember what I said, Sri, it has to be scalable, it has to be replicable, and it has to be integrated. So it also provides very reproducible results. Now, I would honestly say that what I do, like when other people are watching Netflix, I watch GWAS. So GWAS is an association studies where they're talking about genome related data. So as a result, please make sure that you're reading it up on a daily basis. Now, the world has moved really, really forward. In addition to bio conductor packages, so AI can definitely help. Now, Geo is another example where you have gene expression omnibus. Now, all of this data is now available in AlphaFold. So I don't have to go to multiple places. It is like a chart shop. You know, there the bhaiya is going to come and give, and that bhaiya is called AlphaFold and DeepMind. Um, Best practices I already showed: cancer research, genome work, biomarker discovery. Biomarker discovery is very, very important. In fact, right now, we've just kicked that off in the month of August. If you remember, PM Modi has said about diabetes in India. So that is one of the largest biomarker researchers are right now under. There's of course cancer, so that we have biomarkers already. This is standard, and studies are available in DeepMind. In fact, I encourage you to go to OpenAI GPT-5 and ask about AI-driven biomarkers for oncology, specifically to which organ, and you will get it. And by the way, this is me in cyberspace. Uh, Now, later on, this is exactly how the COVID virus looks like. So, we want to bridge between discovery and care. We don't want to make it an academic sort of a setup. So, first is ask yourself 5 questions. Okay? This is all about five or six things that you have to remember. No rocket science. First is start from the bottom. Is it fit for purpose? Is it fit for scale? Is it fit for which phase of the project you're in? Second is, what research are you doing? Will it be only research, or will there be a product at the end? What sort of data type are you having? Secondly, is that how much of compute and infra do you have? And how much of tech expertise you have? The lesser the tech expertise, the better it is. You want more AI that is plug and play, drag and drop sort of mode. So you know which tools to select. And then finally, more importantly, I cannot skip through the details, is that what are the golden nuggets here? There's of course AlphaFold, and this is after you know your basic to gene mapping and coding, right? One of the biggest problems, and I'm taking a minute extra, is the data bias. Remember at the beginning of the call, I told you that AI hallucinates the maximum in two areas. One in biology, pharma. They give you rubbish use cases which you'll think as true, so you have to validate that. And secondly is astrology. So you always need human insight, and human insight is who? You. There's also overfitting problem. That means uh, when the data set is given to you, it works very well, but moment when you are trying to get new real time data, it fails. That's why I said that first, when you have that huge 20 GB of data coming in, make the system run it so that the system gets aware, and that we call as reinforced learning. Now, more importantly, which I think everybody wants to say, that how do I enhance my employability in biology? Now, on this left side, we have our bioinformatics tool, some of the tools that have been very correctly mentioned here. You also have to professionally network. You cannot be an island, right? And then there should be biological data experience, which you can get from your GWAS or genome code. And if you are wanting in today's world or having a career for the next 20-25 years, you need to have a degree or some sort of professional certification in bioinformatics or computational biology. Because if no, if you don't know what linear um, correlation is or linear algorithm is, then I think there is going to be a bit of a problem. But at least for the first 5 years of your career, following your domain bottom up, that is going to make you the top 1% and highly demanded people in the entire world. With that said, I would only say that life is all about smarter research, you knowing your data, making sure you have future researchers and getting plugged in so that you are aware. It's like what you would do to stay in line with Instagram or on Facebook or on Twitter or Gmail or LinkedIn. That's the same thing that you have to do. Baki sab Shri ki Ichha. So I hope I've given you a very, very practical example of how to go about it, how to get started with or without help. What are the basic things that you will have to need? And then finally, how to be more employable. How long does this journey take? This journey takes around 6 months. Mota mota to get really, really hands on. It took me longer because probably it didn't have those same tools, but I assure you it is not as difficult as it seems to be. With that said, any questions, please ask. Sorry, ma'am, I took a little bit more time. It would probably help make things easier.
Definitely, ma'am. So thank you so much for that very much interesting and insightful session, and for sharing your expertise with us today with all those practical real life examples. It was quite relatable for all of our audience, I'm sure.
Ma'am, I would honestly say, please, one thing, sorry. I'm not sharing my experience, I'm sharing my tragedies. Because what I didn't tell you is that after doing 17, 18 months of research, I'm standing in a room, right? I went abroad, I'm like, they got me a business class, everything. And they're saying, okay, you did research. The next thing I think my next colleague will tell, so what? It was shocking, because I was thinking I'm having a Swadesh moment. I have killed myself, literally, my team. But unless and until you show potential, and especially with all the tariffs going on, there's huge pressure in this industry to show output in the shortest amount of time without killing anybody or losing the FDA license. Please believe me, this next 6 months, what you learn through this eminent group is going to be very essential for you to land your career for the next 5 years. That's how it's going to be. So I'm sharing a very vulnerable moment of myself. Don't do the same mistakes that I did, so that you don't have to fall down and learn. You can learn on your own. Urmila, ma'am, I'm
Absolutely.
going and giving it out to you.
Yes, yes, absolutely, absolutely, ma'am. So, if we don't have any questions from the audience, because I think as of now, the audience was so engrossed in the session that they are not realizing that they have to send a question. Maybe they are waiting for the Q&A session at the end. But since we are a little bit short on the timing, we would have to go on to the third session. Although I do have one question from Lakshmi Balasubramaniam. So we will take up this question, Nupur ma'am, and then we will move on to Prodyot's session. Is that fine with you?
Yes, yes, please go ahead.
Yes. So this is the question from Lakshmi Balasubramaniam. Ma'am, I'm full-time working professional from a different field, interested very much in computational biology. How would you advise me to upskill myself in this field?
Yes, very simple. I would say very, very simple. First of all, is that in computational biology, there is a lot of calculations involved in terms of, genomic data, and then especially, I presume you're doing it for medicine. So we have, in addition to what I have explained, there is one. In fact, my eminent colleague would say that in IIT Madras there's a very renowned professor called Dr. Karthik Raman, He's written this entire book on computational, uh, competition system biology. Now, I would suggest that I particularly liked what he had written on metabolic modeling. Get the NP TEL certification. That certification is just 1,000 rupees. Next is, if you really want to get, like, I want to really understand the bioinformatics course, then if you can go to Coursera, there are courses on genomic data science from Johns Hopkins University. I kind of learned from there, and they also tell you about ML and protein folding, and that's little heavy on the coding, but I guess you can do that if you feel very comfortable. If you want something, ki, boss, I don't want to coding vendor. So in that case, go to edX. There is something called as AI for biological sciences, and this is from institutions like MIT and Harvard, so it's really nice on your CV. Go to edX, that's ED and a X capital, and then you can do introduction to computational biology or AI for biological sciences. I think that should do the trick. Last but not the least is that if you really want something awesome, then you have Biotechnica as well for the industry training, and that should do it. The highest and the greatest god of, or we call it the Talaiva of all AI sciences, is Queen Mary University of London, where you have AI in biosciences. It's an online module as well. Cost of bit of money, but that's how you progress. So first, I would start with edX, and then appear for the NAPTEL India Competition System Biology course. And all of this, my friend, is that you can do it while you're working. Because I know I'm a working mom as well, so I constantly keep learning, make good food for the family, and then while they're busy, I can come and do my studies. So, Lakshmi Garu, if that is okay for you, then please proceed ahead. Probably we can connect on LinkedIn, and you can tell me the good news. It works for
Thank
you?
you, ma'am. Thank you so much, ma'am, for guiding all our audiences. So if you would be able to be there till our Q&A session, then um, we
May I
can
go
take up
off
more
camera? May I
questions.
go off camera and grab a
Sure,
glass of
sure.
water? Thank you.
Sure, ma'am. Thank you.
I'm around. I'm around.
Yeah, and then so we'll take up a few more questions at the end in Q&A session. So as of now, let's start off with our third session. I welcome Prodyot sir to kindly join us, and he will be guiding you all how to make the transition into the data science. Prodyot sir,
Yeah,
over to you.
yeah. Thank you. Thank you for having me. Good evening, everyone. So hopefully I'm audible. Can you just confirm?
Yes, sir, you're audible.
Yeah. Okay. Thank you. So hopefully my slides are also visible to everyone. Please
Yes, sir, it's
confirm if
visible.
it is. Okay. Thank you. Yeah. So good evening, everyone. So I just was reading a particular question from Harsha. She was curious about how to do this. Okay, so first of all, I like to thank all the speakers who have spoken before me. Okay, and so basically, her question was that how to do this particular transition. So we are hearing about this. Okay, so how to become a data scientist, transforming from your biology, or you are a wet lab person, you want to transform your career to your bioinformatics or data driven science. Okay, so how to do that? So this particular session will provide you insights on that. Okay, so I will just give you a what is a hawkeye view of what are the things that you need to keep in check. Okay, so obviously we have our courses for that, for the detailed discussion. So but in today's session I'll be giving you a holistic perspective or a bird's eye view, as I said. Okay, so you can follow my like laser for the reference that which particular part of the slide I am talking about. Okay, so basically this particular session caters to how to transform your career from like your biologist to data science. Okay, so first things first, you need not to transform as in like it is not different. Okay, so people might be thinking that like both of the sects are so much different, how can we bridge a link between the two? Okay, so that is where I will try to like tell you that how to build a relationship between the two, what do you say, your fields. Okay, so nowadays we have a whole lot of your interdisciplinary subject. Okay, so one of such subjects can be your bioinformatics, where we are dealing with your like biological science as well as information technology or computational science. Okay. Likewise, there comes data also. Obviously, we are handling a whole lot of data. So we'll talk more about data as well as how to proceed with the data. What are the initial things you need to learn as a beginner. Okay, so basically this particular thing will tell you how to streamline your journey, okay, from day one to how you can transform your like career, and likewise in 6 months or 1 year, and likewise. Okay, so in that particular process we can help you in, and how we can help in. So I will let you know about the same. So this is myself, Prodyot Banerjee. I'm a biotechnical CRO scientist over here. So without further ado, so let's get started. Okay, so first question is like how to like your like this particular convergence happens. Okay, so why biologist basically needs to become a data science, or basically to transform. It is not that you need to transform like fully. Okay, it is not like that. If you are a wet lab scientist, you are, or a wet lab researcher, you can like pursue your own field. But alongside that, let's suppose you are generating a data. Okay, so you are doing your genomics, transcriptomics, proteomics, epigenomics, or your like metabolomics, whatever, in any kind of like what do you say, omics level you are in. Okay, so you are generating a whole lot of data from your wet lab experiments. So if you are by yourself capable enough to analyze that data, okay, so what's better than that? Okay, but because you need not to go to anybody else to like get that particular data analyzed. Okay, so that would be of great help, isn't it? Like you are doing the experiment, you are analyzing the data, you're all sorted. Yeah, so likewise, so that particular thing you have the virtue to do. Okay, but how? So let's talk about that. So first things first, as the first speaker also talked about, there is a whole lot of data, okay, so that we need to basically analyze. But before analysis, basically we need to transform the data into meaningful like insight. To get meaningful insight, basically we need to see that what is the quality of the data. Okay, the data is well managed or not, because you see the raw data is like going here. Okay, so it is not in a particular format, so you need to format that in a particular way, how you can analyze it further. Okay, so that is the first challenge that comes across. Okay, so when we talk about the data in biological data, what kind of data we are talking about? We are talking about all sorts of data that the biology generates. Okay, be it your next gen sequencing based data, so be it your like first gen, second gen, third gen, whatever sequencing based data, or you are talking about multi-omics data. Omics basically means to study. Okay, so whatever you are studying, like if you are studying genomics, it is like genome, basically it is genomics. That you are studying about transcriptome, it is transcriptomics. Likewise for your proteomics and your metabolomics. Okay, likewise. Why? Because we need to understand the data at every omics level. Why is it relevant? Because see, so there is a whole cascade of things that you see. Okay, so if I just have to give you an example, let's suppose you are talking about a wall. Okay, so a wall is made out of bricks, isn't it? So if you can analyze the anomaly in a brick, okay, so you can like basically analyze that where the positive agent of that particular thing is. Okay, so that is what I'm talking about, genomics. If I can identify a particular variance at the genomic level, it will give me a power that how the particular thing will further proceed in the what do you say, your proteomic level or your metabolomic level, or the cascade of reaction that follows. Okay, likewise. So obviously we know that all kind of variances are not gonna cause causing a kind of mutation, or some of the mutation can be silent, does that. Okay, that is there. But we are basically dealing with a whole lot of data. So that is the main thing, because NGS also what do you say generates GBs of data. Okay, we are talking about like gigabytes, terabytes of data. Okay, so massive amount of data obviously needs to be catered. Okay, and for that particular thing, we had bioinformatics, and nowadays we have obviously your data driven science, or you call it as AI, which can automate the things. Okay, so because see, manually doing the things now in today's generation doesn't make sense, isn't it? So you need to get your computer doing the things for you. Okay, just like Warren Buffett says that if you are sleeping and your money is not working, so you are doing no good. Likewise, in today's world, if you are doing manual stuff, okay, it doesn't make sense. Okay, so like you should be developing an algorithm which will be doing the job for you. Okay, so basically complete automation we're talking about. Okay, but obviously that is a like jargonish word for a a person starting in this particular, what do you say, domain. Okay, so we need to understand where to start from. Okay, so that is where I'm just trying to convey that particular thing, how to start. Okay, so initially what we had is the traditional analytical tools from the likes of your Excel or basic statistics that you have. Okay, so but that particular thing doesn't hold true. Okay, in today's world. Okay, obviously the basic remains the same. So, okay, same. It is not that the basics are gonna change. Okay, in other terms, if I have to quote, the AI is just a fancier version of your statistics only. Okay, so basically you have to start from absolute basic to know what is statistics. Okay, so how we can analyze the data, what is the data all about. Okay, so for that particular thing you need to have at least a what do you say, your bachelor's level biology knowledge at least. Okay, then you can build upon from that. Okay, so what is our main goal over here? So basically we are trying to find patterns. Okay, so our previous speakers have also talked about how to find patterns. So in AI world, finding pattern is a very relevant thing. Okay, so you talk about convolutional neural network, it can find patterns in your image based data. Let's suppose we are talking about your CT scan, like PET CT, MRI, CT scan, or any kind of image based data. Okay, so now you see that we can detect a like tumor of breast cancer 5 years before it's like, what do you say, your like it has grown in masses, or like it has become, what do you say, your like transformative, or it has become like a third stage cancer or this that. Okay, so we can basically analyze it, or basically what do you say, find the pattern of it and try to see that, okay, so this is the positive agent and how we can work on this particular thing. Okay, so likewise this particular thing, like if I have to give you a generic example from the likes of your Google Lens, also works in this particular thing, that you click a snap and it will try to like find the pattern from that particular image and show you around that where it can find similar images. Okay, so like you are like taking a snap of a bag or something, or a flower. Okay, so it can give you that what species it belongs to. Okay, so where you can find it, and likewise. Okay, similar things we can do with our medical data as well. Okay, so I have given this particular example so that everyone can relate to it. Okay, likewise. So then talking about your like data science based skill. So why is it important? Basically, if you have the data science based skill, or basically if you can integrate your biological knowledge with your computational knowledge, what happens is you can basically work with any kind of data which is belonging to any kind of interdisciplinary subject. Okay, or you can handle multiple interdisciplinary project. Why does that matter? Because nowadays whole lot of disciplines are also coming in. Okay, as you understand. Okay, so the world is getting more specific, to be honest. Okay, so for that particular thing, a whole lot of interdisciplinary subjects are obviously turning up, and in order to do that you need to always be learning. Okay, so first and foremost thing is the AI is obviously exponentially growing, and you have to be in that particular pace to catch that. Okay, so that is the main objective. Okay, so you always have to be on your like platforms like a GitHub or Kaggle. Okay, so you need to like what do you say, be in touch with the experts. Okay, or you can like take a what do you say, upgradation course. Okay, so to keep a check that you are following the industrial standards or not. Okay, likewise those things. Okay, so apart from that, like as I said, that you need not to transform holistically, that you just transform to a fully data scientist. Okay, not required. Okay, so you can be a lab driven scientist. Okay, having some knowledge of your software what do you say, skill sets, where you can analyze your data by yourself without needing the assistance of any other. Okay, apart from that, if you want to design a particular algorithm to have a better work on your particular kind of data that you cater. Okay, apart from that, you're translating biological findings for your any kind of actionable insight. Okay, so basically the main purpose of the research is what? Okay, basically whatever agent you're working upon, you want to get some actionable results out of it. Okay, so that is the main motive. Okay, so likewise so then coming on to your career opportunities. Okay, so obviously for a beginner, everyone is looking for job prospects, isn't it? So if you go through your like any of the job posting sites from the likes of Biotechnica is also one of them. Apart from that, LinkedIn is there. So if you see that most of the job opportunities that you can find in here is obviously NGS data pipeline automation using AI. Apart from that, drug discovery scientist that you have, bioinformatician, okay, goes without saying. Biomarker analyst, okay, so if you are trying to come up with a biomarker discovery of your complex disorders like your cancer, neurodegenerative disorders from the likes of your Parkinson's, Huntington, or your amyotrophic lateral sclerosis, and number of such disorders. Okay, it can be. Okay, then your research scientist, computational research scientist, or in silico scientist, basically, okay, dry lab scientist. So that is there, data curator, or you can be a full stack developer. Okay, so or you can be a back end, front end developer. So developing what is the softwares from the likes of your softwares like your PyMol, Rasmol, or any kind of visualization tool, or your GEO, the thing that ma'am has already told you. Okay, so either you are going for a visualization tool or a data transformation tool, or if you are coming up with a new kind of an algorithm, like see, choosing the algorithm is very much important. Okay, so I can tell you about random forest or CNN, RNN. Okay, so that is there, but obviously which one to choose in which use case. Okay, so what is the use case of a particular thing? Like RNN would be more fruitful when you are dealing with the sequential data, whereas CNN would be more fruitful when we are dealing with image based data. Likewise, you need to understand, okay, so the use cases. Okay, so likewise, so for learning pathway, so basically work smarter, not harder. Okay, so we have seen from the lifelong that from our childhood we are seeing that, yeah, work hard, work hard. You can't work harder than a labor. Okay, so obviously, or I'm not demeaning anyone. I'm just trying to say that you can't basically do a hard work you can't compete on hard to everyone, but you can compete on the smarter front. Okay, so likewise, you should be doing the things in a much smarter way. It is just like a mathematical problem can be solved in a number of ways. Okay, as you understand. Okay, so someone would be doing it in, let's suppose, 9 steps, someone would be doing it in 2 steps. Okay, for a beginner, it is obviously important to do it in 9 steps, because you will be understanding what are the, what do you say, things that are there. Okay, so each and every step. But if you become a pro, obviously you can do it in 2 steps. Obviously that is the smarter way to do it. Okay, likewise, now coming on to essential tools. Okay, so let me get a uh, pen instead. Okay, so yeah, so basically let's talk about the essential tools. Okay, absolute essentials. So I hope that some of you might not be aware of this particular, or you're not versed with R or Python. Okay, but you must have heard of them. Okay, because these are industrial standards nowadays. As you understand why we are talking about Python and R, there are a whole lot of languages like your .NET, then C, C++, Java, this, that. Okay, so likewise. But why we are talking about Python and R when it comes to your biological data? Because the libraries in your Python and R supports the cause. Okay, so basically when we talk about your packages like your scikit-learn for your training, for your machine learning based algorithm, then your TensorFlow is there. Apart from that, you have your BioPython package from your Python. Okay, so which caters to your biological data analysis. Okay, so you talk about sequence IO, that particular thing you won't be finding in some other languages to read the sequences, and likewise. Okay, so then obviously automation, machine learning, everything it is capable of, all other languages are also capable of. But why we are focusing upon Python and R, the main and absolute reason is here that the libraries over here caters to the biological data from the likes of your, be it your ggplot for your visualization, okay, or plotly for your visualization, from the bioconductor packages in R. Okay, so similar things actually you can find in both of the languages. For example, if you talk about matplotlib or your matplotlib in Python for your visualization, same thing can be done using ggplot in R. Okay, so likewise you have your bioconda and this bioconductor in your R, then caret, this, that. So multiple parallel libraries you can find in both of the languages. So I would emphasize that if you are a beginner, okay, learn one of them. Okay, so no need to jot your mind on both of them. Okay, so if you are trying to have a proficiency, if you are having a proficiency in either of them, that will do, that will sail your boat. Okay, no need to jot your mind in trying to understand both of them. Okay, but having said that, obviously your logic of the coding remains the same. either it whatever be the language. Okay, so the logic of the coding remains the same. It always starts with the header. It will have a body. It will have a return function and likewise. Okay, so yeah. So now coming on to a very basic question that we encounter time and again. Okay, so that is how much of a maths is really required. Okay, so because being a biologist, many of you, I'm not saying all of you, but many of you are very frightened of mathematics. Okay, and when we talk about data-driven science, obviously mathematics is the key. Okay, so how much of a mathematics is required? Okay, so actually you don't require or what do you say, whopping amount of mathematics or likewise nothing of that sort. Okay, so you just have to have a fundamental understanding of the things. Okay, so everything should make sense to you, that much only. Okay, so talking about statistics, the basic statistics of your hypothesis testing. Okay, so null hypothesis, alternative hypothesis, does that, p values, or what is the p value, what is the significance value, why we do it as 0.05 or 5%, why we take. Okay, what is the regression? Okay, what is the linear regression, what are the types of regression, why regression is dynamic in nature, and this that. Okay, then coming on to your basics of your linear algebra and your like calculus, the basics of your what is the gradient, what are the matrices, what is the 2 into 2 matrix. Okay, so this that are a basic of understanding of that will say you good. Okay, so no need to be an expert of mathematics for that matter. Okay, now coming on to critical mistakes that you should avoid as a beginner. Okay, so always we're talking about beginner over here. Okay, so basically don't jump onto like the harder part like deep learning. So deep learning is far more complicated if you compare it to any of the like simple machine learning or your like any part of deep learning, be it your neural networks, is that. Okay, or NLP. Even NLP is what do you say, kind of a easier thing. Okay, so basically the main gist of the thing is don't jump onto the harder part straight away. First understand what is the supervised learning, what is the unsupervised, what is the like recurrent neural network, reinforcement learning part. Okay, so what is the reinforcement learning, and likewise, okay, how does the reward work, okay, how does the penalty work, why we are penalizing it, this that. Okay, so first be a master of the easier thing like data cleaning. Okay, so very basic data cleaning, but it is basic but very important. Okay, so if you are catering to the raw data, if you haven't cleaned your data, or data wrangling as we call it as, okay, if you haven't done that, it makes no sense that you run a deep learning algorithm upon. Okay, so exploratory data analysis, basic statistics. Okay, so the basics you have to be very clear about. It is not only about data science or anything. In biology also you always have to be very clear on basics. If I have to talk about basics of biology, if you are not good at microbiology, you are not good at biology at all. Okay, so mostly, okay, so I'm not always emphasizing, but basically obviously all kind of molecular biology techniques, be it your multiomics that we are talking about, everything comes under that. Whatever we are talking about, genomics, be it your transcriptomics, epigenetics, proteomics, omics, metabolomics, everything comes under that. Okay, likewise, so the simplicity is the key. Okay, first be clear with the basics and then move on to the further part of the things. Okay, and don't be frightened with the what do you say, your bigger terms like deep learning, this that. You will slowly and gradually learn it. Okay, so don't like be frightened of that. Okay, it is just like why we should like start from your basics. Basically we don't want to do this garbage in and garbage out. Okay, because we need ourselves to like whatever we are learning, it should make sense to us, and we should be able to answer the question why we are going on to neural network. What is the need of the hour? Like why we need a neural network, why the basic algorithm is not able to solve that particular problem. Okay, why do we need GPUs, why do we need NPUs, okay, for this particular problem. Okay, so why the data is like whenever we are dealing with a very integrated data, why do we need a deep learning. Okay, so those particular things will be answered. Okay, so likewise if you join our session, we like emphasize more upon these things, why. Okay, so and science is all about whys. Okay, as you understand. Okay, so now coming on to gaining experience, as ma'am has also stated that in order to gain experience there is no alternative. Okay, so you need to work, that is the only way. Okay, so you need to get started, so that is the first thing. So today is the day. Okay, so there is no tomorrow. Okay, so like obviously you have to get started and you need to do the things on your laptop first. Okay, so basically we are talking about being a in silico based researcher, so you can do all of the stuff using your regular computer or even in HPCCs for higher level stuffs also. Okay, but it would be in your computers. Okay, likewise, so basically build your portfolio. Okay, you don't need a wet lab to basically get started in in silico based research if you are into. Okay, so basically you can sit at the ease of your home and doing your research. Okay, so not always at home, I'm not like telling about that, but you can do that. So that is already a prospect is there. Okay, so you can basically work remotely, and likewise remotely doesn't always mean like you are working from home and ease of it. Okay, but it also means that you can collaborate with n number of scientists abroad. Okay, so let's suppose you're collaborating with US, UK, like we do, our CRO division does that. Okay, so how we are able to crack that? Because we are wet lab scientists, we can handle that particular thing, and you will also be able to do so. So in order to do that, obviously today you can get started with your projects and industrial standard things. What is going on the on the front of industries that you need to learn first. Okay, so with some examples of the project that you can tackle, obviously there are a number of projects that we handle. You can visit our website for regarding the same. But as we are talking about what types of projects that you can handle, any kind of project that is there in your, like from the likes of your drug discovery, then your NGS, apart from that your multiomics level analysis, any kind of analysis that you can talk of. Okay, I have already told you that from basics, if you have to start variance analysis, biomarker discovery at the genomic level, then like your gene expression. Okay, so gene expression analysis, or after NGS also you do differential gene expression as stated by our previous speaker. Okay, so apart from that, drug discovery also comes into picture from the likes of your automation of your MD simulation, docking processes, admit screening, this that. Okay, yeah. Then coming on to your end-to-end skills that you should learn. Okay, so like study designing and data acquisition like from the likes of your GEO, that is gene expression omnibus, then your SRA database is there. Okay, if you are catering to the cancer based data, then TCG is there. Okay, so like the Genome Atlas. Okay, data wrangling and cleaning. Okay, so that is the main work. That is what I said. It is basic, but it is very important. Okay, apart from that, your algorithm selection and application, as I have already stated, n number of algorithms are there, but what fits the what do you say, your particular thing that you want to do, based on that you will be choosing. Okay, so every algorithm has its boon and bane. Okay, it is not that none of the algorithm is like foolproof. Okay, so it depends upon the use case always. Okay, so interpreting and storytelling, it is very important to bridge the link between all of the things, so that you can basically come up with a like streamlined workflow of the things. Because whenever, let's suppose you are writing a paper, okay, so paper is nothing but a story telling what you did. Okay, so it is not that taking some things from here and there, like putting the references and quoting a story. It is not like that. Whatever you have done, it should be replicated well. Okay, so it should be what do you say, translated well to the readers. Okay, so that is the main purpose of a researcher while writing a paper. Okay, obviously novel findings comes into picture, but obviously if you are following a streamlined process, you have the most amount of probability to land up with a novel finding. Okay, so yeah, likewise. So like how can you get the direct experience? Basically remote internships that you can do. Some of the platforms ma'am has already named, like edX is there, Biotechnica is obviously there. Okay, so likewise you can also get your internships from the research organization, from the likes of your TIFR, IITs, ISCs, then your likewise all of the central government research institute. Okay, so CSIR, ICMRs. Okay, Tata Memorial, there's that many others, many of them are there. Okay, internationally also we have Stanford, Harvard, there's that. Okay, so then you have like industrial based, pharma based, like Pfizer's and like Novartis, like you name. Like apart from that, in silico, if you're talking about, you can go to Schrodinger. Okay, so pioneer company in their drug discovery. So then your Agnita AI is doing great job. Okay, so based out of your Bangalore, Hyderabad. They have their head office in Hyderabad. Okay, so then you have research roles in your computational biology groups. Okay, so talking about your from the likes of your GitHub, you can go on to Kaggle, you can go on to, obviously. So networking is very important. Okay, strong applications like from your masters, either you are a master, PhD, or like degree does matter. I'm not saying that. Obviously, degree does matter till the level you are like qualifying the HR round. This that. After that, obviously, your skill takes over. Okay, so without a skill, with only a degree, you can't go forward much. Okay, so that is there. So your journey basically starts from today. As I said, that it is always day zero or today, because there is no tomorrow. So basically, so what is the summary? So basically, the summary is your domain expertise, as like Nupur ma'am was also emphasizing upon the same, that your domain expertise is absolutely integral. Okay, so without a domain expertise, you cannot move forward too much. Okay, so that is why a bridging link between the data science and biology is very critical. You can't be, what do you say, expert of one of the things, or you can be jack of all trades. That is okay, but likewise, you need to integrate both of them to have meaningful insights. Okay, so combining the both your like computational knowledge alongside your what do you say, biology knowledge. Okay, so biology can be any kind of biology. Okay, so you can be from microbiology, or you can be from your molecular biology, or ecology. Anything will do. Okay, so but you need to figure out that how AI can be integrated to your particular data. Okay, so likewise, so focus on the fundamental statistics. Okay, so coding, the basics of coding are basically Python and R. Okay, for a beginner. So now one might ask that what would you suggest, that Python or R? So it basically completely depends upon your what do you say, like user preference basically. Okay, it is not that I find R easier, or like someone might find Python easier. So it is like that. Okay, so both are like easier as compared to all other medieval level languages that you find. Okay, so practical projects that you can do. Okay, so in that particular tangent, Biotechnica can also help us with. Okay, so we are handling a whole lot of projects. Okay, international, national, and of industrial standards as well. Okay, so choosing the language. Okay, so I'm just go quickly on this because I will not take up much of your time. Already taken some part of time. Yeah. So basically, choose your language, whatever like language you're comfortable in. If you want to learn, if you get started, so Biotechnica is the platform, edX is there, GitHub is there. Okay, Kaggle is also there. Okay, apart from that, finding a data set. So like repositories are always there. Okay, so freely data is available. Okay, so TCG is there for cancer-based data. NCBI is always there. Like the what do you say, basic what do you say, your bioinformatics. Your data set is there. Apart from that, you have Geo, that is your Gene Expression Omnibus. Apart from that, Kaggle is there. So there are a whole lot of platforms to get your data from. Okay, raw data, getting a raw data nowadays is not a big deal. Okay, apart from that, you're building your project upon that, what you want to interpret, because the interpretation is the main thing that a researcher is bringing onto the table. Okay, data is always there. Okay, what you want to conclude from there, so that is your part of the job that you are doing. Okay, so what kind of interpretation you come up with, what quality you serve. Okay, and based on that, you will be having the quality of the publication as well. Okay, so apart from that, joining the community, as everyone emphasizes that obviously like you have to join the communities from the likes of your like technical communities, from the likes of your GitHub. Okay, CRAN is another repository. Okay, there's that biostars, or your like Stack Overflow. You can be a part of your like hackathons. Okay, so you can take part in hackathons, this that. Okay, likewise, apart from that, obviously social media based interaction is also helpful, like from the likes of LinkedIn, this that. Okay, so final thoughts. Basically, whenever you are starting something, you don't think that you are starting from scratch. Okay, so you are basically powerfully integrating two of the worlds. Okay, so it will take time, but obviously you are doing a noble thing. Like just believe in yourself and get started. Okay, so that is the thing. Okay, with that particular concluding note, I conclude my session over here. So hopefully you have learned something new today. So with that particular note, I will take your leave. Okay, bye bye, and have a nice day ahead to the participants joining from abroad, and good night to the participants joining from India. Bye bye. Till the next time.
Thank you so much, Mr. Prodyot sir, for those very actionable steps on how to start this journey. And the points which you mentioned about avoiding mistakes and working on dry lab projects and to start small was very inspiring. Okay, so with that, we come to an end with all those fantastic sessions. I just want to remind you that your journey can start small, begin with Python or R, take on guided projects, and gradually build a portfolio that can open doors to internships, remote research roles, and higher studies abroad. But I'm sure still many of you might be thinking, I'm afraid of coding. Can I really do this? I don't have a computer science background. Where do I begin? But here's the good news. You don't need to figure this out alone. As Nupur ma'am already suggested, at Biotechnica, we have designed two student friendly programs that will help you start with coding, that is coding for biologists, and eventually build advanced data science skills. So we are starting our course on data science hands-on internship for biologists. It's a 120 days training with everything which is incorporated in it. It starts 18 September. So all while working on real biology data sets. So these guided internships and project-based courses are the fastest way to apply what you have learned today to your own research or thesis. So if you're serious about leading your research like a pro, then this is your perfect starting point. So since we have already exceeded one our time, and Dr. Nupur ma'am has already answered one question. So we have one more question which I would like to bring up on the screen. This is from Zahia. What do you think about the future biology and AI? Nupur ma'am, we would love you to answer this question, please.
Okay, what is the future of a calculator on your desk and you doing maths? Same answer. You cannot, we are living in a world where you cannot separate AI from biology, right? And as Dr. Saab said, that the future belongs to those who use AI and other people who are using AI for Ghibli pictures. So now you have to understand that what you are using AI for. Otherwise, like you've seen some of the movies, right, Zahaya, कि people are pointing guns at you और आप kung fu कर रहे हो, मर जाओगे आप, right? You know that it's not that Bruce Lee did kung fu, so I'm going to live. Nah. You don't bring a knife to a gunfight. So here, what Biotechnica is saying is the right thing to do is because when you are coming to a gunfight, you have to know some of these basic fundamentals by which you know that AI is like a supermarket. You need to know which counter to go to किया डिटर्जेंट का काउंटर है कि फेस क्रीम का काउंटर. You should not end up like I ended up there was in a Switzerland place and I couldn't read the data what was written. It was dishwashing liquid with a very beautiful blonde lady on top of it, and I thought that it is for hair. My hair साफ हुआ कि नहीं हुआ, but बर्तन जैसा चमका है. So I'm just saying that you have to know some of these basic ideas. Biostatistics is AI is already there. Second part of it is protein strain detection. AI is already there. And in addition to what other things that are there in AI, which is probably not in Biotechnica, is that see one of the most painful and a lot of waiting and wringing your hands sort of setup is getting the approvals from FDA, submitting the regulatory documents where you have to validate and test and assess, and then is your model getting fitted, is the data correct or not, do I need always a wet lab to get things forward. So a lot of those trials and paperwork that is required, including if you buy a medicine, you remember that small chithi or the prescription document is given inside the bag. That, do you know something, if a drug takes 2 years to make, it takes 7 months just to decipher what I'm going to put in the literature, because that goes to multiple regulatory approvals. In those areas, AI is used for automation. But yes, it is supposed to reduce the grunt work. Future क्या present है, it is like you have to learn in order to be the future, नहीं तो future नहीं है. So I'll be very very blunt with you. So you can do maths on your own, but there is always a calculator. So next time you think about this question, say what Excel is to your computer, AI is to your future biology. I hope I've answered that. No pun intended.
Thank you so much. Thank you so much, ma'am. I just simply love how you can relate so quickly to real life examples, and that too in such a
Doctor, no, I would ask Doctor
amusing
Sir
note.
to also give him, chime in. If I'm giving an industry example, but from a professional point of view, Doctor Sir, I would really like you to also give your perspective to it.
No, you're absolutely on point, obviously. So, yeah, so obviously we are already in the future, and as I have already quoted, that it is like growing exponentially. So, obviously, you have to be like keep pace to catch that. If you are like like what is about. So future, if you're talking about what do you say, 5 years back, we haven't thought that something like DeepMind will come and have something like your AlphaFold or your AlphaMissense for your variance analysis, for your protein prediction. Okay, so obviously, so it is going fast, and I always say this, like with the advent of more advanced computational, what do you say, like GPUs and NPUs from NVIDIA, particularly, if I just have to name a company. So obviously, we are going better. So yeah.
Yeah. So thank you so much, Pradut sir, for that insight. And with that, we will have to end the session over here since we have already exceeded our time. I would like to remind our audience that if you want to connect with Dr. Nupur Mukherjee ma'am, then you can definitely connect with her on LinkedIn. If you have any more questions to her, ma'am will be more than happy to reply you back. Okay. So, with that final wrap up, I would like to express my heartfelt thanks to our wonderful speakers of today, Ms. Shubhi Singh, Dr. Nupur Mukherjee, and Mr. Prodyot Banerjee, sir. Thank you so much for your sharing your experience and inspiring our audience today. And a big
Thank you,
thank
everyone.
to and a big thank you to everyone who joined us. Remember, biology plus data plus creativity. It is the formula for tomorrow's discovery. So start small. You can definitely manage the crash course which we were talking about. Sumit Kumar, you have asked how to manage both college. So these sessions happen during in the evening time from 7 p.m. onwards. Okay, so this would be very easy for you to manage, and I'm pretty sure that. You can manage your college studies during the weekends also if you attend these course sessions in the weekdays, right? So start small, stay consistent, and you'll be amazed at how far you can go. So with that, have a great evening, and we will see you in our next session. Thank you so much, everyone.
Thank you, everyone.
Thank you, everyone.
Thank you, everyone. May you be the future leaders that I get to work for and work with.
Absolutely. Thank you, ma'am.
Thank you.
Good night.